ये था ना हाथ छोटा का कलर विचार है क्या है इसका लीवर है लीवर
मैं तो दिखती नहीं उसे भाई देखते हुए हम लोग प्रोजेक्ट के काम में मेरे प्रोजेक्ट कंप्लीट किया से मेरा कर रहे हैं अभी जो मेरा बना है नहीं या ये नहीं ये प्रोजेक्ट इंस्टाग्राम पे पोस्ट करते हैं इसके बाद बहुत चाहते हैं बहुत ही लोगों को लगता है कि ये मैथ है ये बच्चा सबको लगता है नहीं है शक्कर देखो यार इसकी शक्कर देखो कितनी अच्छी मेहंदी लगा दी थी
एक क्लास में गरम नहीं रहता एक क्लास में गरम नहीं रहता
भाई मैं आज से
दिखा
ये क्या है
हाथ तो मिला हो क्या
पहले
रोले
ये 6 महीने की थी मतलब का है
तो फिट का कर रिलेशन
कितनी चालू बच्ची थी देखा है क्या
फिट है अभी भी ऐसी है
ओ
नहीं तो चलो नहीं होता भाई
मैं तो भी नहीं
अबे मैं क्या हुआ शायद थोड़ा मैं लक्ष्मी को हाथ में बोल रहा था लक्ष्मी दिया हाथ
लक्ष्मी कौन है
अरे बॉम अबे
नहीं हुआ
मैं हाथ से मैं फोड़ के फेंकने वाला था पर वो हाथ में फूट गया बॉम पूरा मेरा पूरा आँख में सफेद सफेद दिखने लग गया पूरा कान सुन्न हो गया जैसे फ्लैश बैंग डालते हैं ना धक्के से पूरा अरे हाँ प्लीज ना मैं कितने दिनों से गेम नहीं खेला मैं आज से
अबे अभी डॉक्टर नहीं हो रहा कुछ भाई
कभी कुछ भी
हां अभी 2 महीने पहले बहुत ठंड लगने के बाद क्या रेडिएशन हो गया क्या भाई मेरे हाथ में
भाई मेरा भी
मेरा भी हाथ में
हमारे कितने पागल होते थे वो लोग लेके ना सब फेंक देते हैं
क्या
मतलब वो चलाकर दूसरों में फेंक देना
तो मैं भी कैसे
ठीक है
अभी मैं घर जा रहा था पर मोरट में किसी को भी फेंक दिया सीधा मेरे सामने
मुराद में कौन
इसने मेरे हाथ को पूरा जला दिया
अच्छा सॉरी
वो भी करता है ना वो भी शूट
ठीक होता है
बहुत मेरे को पूरा फुल गया था
अबे छी भाई मैं बोल रहा हूँ मेरे हाथ में लक्ष्मी को फूटा है पूरा आँख सुन्न हो गया था निकाल सुन्न हो गया
वो इतना आवाज करता है वो इतना नहीं फोड़ना चाहिए
वो पूरा बॉम्ब था
मैं दिवाली पे घर से
वो ऐसे बॉम्ब होता है ना
फोड़ा हुआ बॉस
सही में
आवाज आ रहा सही में
बट फूटा नहीं
मैंने इसे फेंका और फूटा नहीं बट वो बाहर फूट गया
अबे बॉस में
अच्छा विंडो के बाद अरे मैं
अबे किसी के ऊपर गिर गया
भाई एक मिनट देखो ना कितने फोड़ना आता है वो भी नहीं इससे बिना फोड़े फेंक दिया
मैं छी भाई
उसको आता ही नहीं
मुझे आता है वो मैं कल लिखा था
अबे मैं हमें पढ़ना करता हूँ अभिप्राय
ये ऐसा ऐसा क्यों होता है
भाई वो ना उसको हेड पता नहीं
अरे मेरे को भी नहीं मिला
अबे सुन मैं कल असली वाला मान लेता हूँ मेरा वाला
हाँ वो
देखो पता है
वो डब्बा भाई
हां याद है नहीं देख भाई
अबे
बहुत गिर गया
बीड़ी बॉम्ब
अबे
अरे इसका हम चाइना का मारा
बीड़ी बॉम्ब
नहीं मेरे लिए
हम मिलेंगे
अबे मैंने वो चाइना जैसा
और उन्होंने वो रिएक्शन दिया
फाइनली
पहले से लाइफ में था आपको देखा नहीं
लेकिन बहुत सच में बिल्कुल आपने
ऐसा काम तो कभी बता मैं बता सपोर्ट कर यार वो कूल जाके कान में
अभी ब्लैक को भी बोला अभी मैंने भी बोला
सोलिका तू तो होती है नहीं भाई अभी
अभी
करेगा ये
मैं ऑक्टोपस में चलेगा
वॉशरूम में जाके
वो सपोर्ट करेगा
मैं ऑक्टोपस
क्या
क्या कर रहा है
मैं क्या
अबे आज तो तुमने स्नैप नहीं भाई
अरे खुद बहुत कम अगर मैं करता तो मैं ये करके दिखाऊं
मैं
मोटिवेशन
ये थोड़ा अजीब अरे अभी मत कर
अभी मैं नहीं करते भाई क्लास में कोई
एक बार
मत
अबे ये डर गया ना अभी सर
आज में लेक्चर चालू है समझ में आता है आपको एक बार बताइए
ये पता नहीं कौन है
अबे नहीं कुछ भी किया
अबे इतना नहीं है
अबे क्या
अबे बड़ा चांस
डॉन
पुली अबे भाई आएंगे पुली बचा रहे हो
मस्ती क्यों कर रहे हो भाई बुलाओ
ये पकड़ा है अबे
क्या है
क्या है देखो मैंने अरे मैं हटो
तू शांत बोल बोली मत
अरे कैमरा चेक कर लो
बाजू में लेक्चर चालू है समझ में
अरे
आ रहा है
अरे
बोलने का लक्ष्य अबे तेरा आवाज क्यों
तू क्या बोल रहा है
कुछ जल्दी अबे
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 क्या इधर
दो बार काम
क्लास में कौन ही चलेगा
रिफ्लेक्शन क्यों किया
नहीं दे
डॉन है ना तू डॉन से थोड़ा सा यहां मेरे को भी दे यार ऐसे कर
एक काम करते हैं नहीं छोड़
बोल
मैं क्या बोल रहा था ना सुना लेते
अबे यार कुछ भी बकवास
उसका तेरा क्वेश्चन आंसर देगा है आंसर नहीं हेडेक होता है एक्सट्रीम ऑफ हॉटनेस में
वो इधर डाल देगा
ये अभी तक निकल चल
भाई मैंने लाइक इसका क्वेश्चन आंसर कर दिया
अभी
तेरे में
तेरे
ट्राई
डालना है
तेरे में ट्राई कर
नो नो
तू टेस्ट सब्जेक्ट है
अच्छा देख क्या बोल रही है
आज तो बोल रही है जी
मैंने आपने बोला तुझे क्या करना
ओ क्या हुआ
भाई इसने बोला
क्या ब्लूपर्स आ गए भाई ये क्या
यार बकवास है
ग्लूकोज मतलब बिफोर सीन्स आफ्टर सीन्स
ओ ऐसा मैंने कुछ नहीं कहा
तू क्या बोल रहा था
बिफोर सीन आफ्टर सीन क्या होता है
अरे देखा था एक अरे डेस्कटॉप तक नहीं देखा मैं देखा
जाना नहीं मैंने क्या लेक्चर से देख लिया
वो ऐसा अपनी मस्ती कर रहा है
क्या हुआ
अरे
बोलना
अरे ग्लूकोज मतलब बेसिकली एंड का जो भी हुआ पूरे शूट में
एवोल्यूशन
तूने दिखाया डॉक्टर
कहां से
क्वेश्चन नहीं होता
अच्छा ब्लूपर
तूने कुछ नहीं बोला तू क्या
नाम सुना वैसे नाम आने का
अबे बात अबे मैं जरा नहीं पूरी काट में जरूर से
तूने सिर्फ तूने काम दिस प्रॉब्लम बस और तेरा पेट सॉफ्ट हो जाएगा
नहीं मैं तो
सॉरी बात चालू
क्या है
सॉरी बात चालू
अभी क्या ऐसे नहीं आता ये
सॉरी
ब्रो का
इट इज अ सेफ मेडिसिन ये नहीं मैं चला
ये अभी कुछ ना
ओए ओए अभी मत चला
विमान
कोई जाएगा
क्लास सॉरी बात
साइड इसका एक दो करता है उसको फेंक देता है
चलिए
डरा मत कर तो कर ही नहीं पाएगा
अभी तेरा बहुत डरावना वाली फीलिंग है
नहीं
बहुत डरावना
कुछ भी बकवास करेंगे
फिर कंसंट्रेटेड
इसमें ईयरबड्स नहीं
फोन नहीं दे रहा
मस्टर्ड
फोन
नहीं
नहीं
मिलेगा भाई
छोड़ देना रिकॉर्ड दे
मतलब आई एम नॉट श्योर ज्यादा डालूंगी मेरे कान में कभी कभी ज्यादा दर्द वो डाल के 48 डालूंगी
ये भाई अंशु सो कर गया
अबे नहीं जैसे लाइक हट के कान में ईयरबड्स डालना होता है वो डराना It will just come out.
But दोनों का mix
अबे आदित्य नहीं आदित्य पट्टू इधर नहीं यार
Exactly.
हमें सब देख कर क्या है क्या बोलते हैं हमें वो मरने के बाद मरने के बाद निकालने
एक मिनट मैं उसको माना कि मेरे को तो नहीं मारता
मैं तो मारा नहीं
मैंने बोला
पहली बात तो मैं तेरे को फेंका नहीं
क्यों आगे किसकी
इधर गया
वो भी
इधर गया और तुम लोग जोर से मारो कितना नहीं बचता है खुद होता है खुद पर से बच गिरा कर समझ गए सबने जगह खुद मतलब कैसे
मैंने
शान सीधा बैठो इधर लेक्चर चल रहा है ना
Next is after the functions, we'll again have to understand what are the other features of testes. So far, the functions of the main system that we had studied. Take one question, number one, first. Number one, this you'll have to try answering. Now that I give you these questions, try answering on your own, write properly. Number one. Number one, name the part of the male system. Name the part in the male system that transfers gametes from testis to urethra. Keep a gap for the answer. Number two. Number two. Write two special features of the male gamete. Write two special features of the male gamete. Write two special features of the male gamete. Special features, I'll tell you. Space rakho, go to number 3. Go to number 3. Name the copulatory organ. Name the copulatory organ. Name the copulatory organ. One line for the answer. Number 4.
Those special features or feature
What?
of the same question. Special features of male gamete.
Special features. Two special features of the male gamete. Name the copulatory organ in the male body. And number 4. Number 5.
मोटाई
5 now. Number 4. Okay. Number 4. What is the significance of the membranous sac surrounding the testis. Significance of membranous sac surrounding the testis. This answer you write down. This comes for 2 marks also, as I said. Either this or this. Answer is almost the same. Write down. This answer you will write down. I'll dictate. Likho. Significance of the membranous sac surrounding.
It's contraction, relaxation, temperature.
That's बहुत अच्छी तरह से लिखना है. Write down number. Write down the answer for that only. The rest you will try. The membranous sac surrounding the testis. Likho. Membranous sac surrounding the testis is known as the scrotal sac. Full stop. The scrotal sac contracts.
This is the question.
Significance of scrotal sac. Significance of the scrotal sac. Whether you are asked this or this, answer is almost the same. Then the scrotal sac contracts and takes the testis towards the body. Takes the testis towards the body.
My class is still 5:55, 5:40.
Now you write your answer. Now you write your answer. Takes the testis towards the body to maintain a temperature suitable for sperm formation full stop. This happens when the outside temperature this happens when the outside temperature is much lower than body temperature.
Ma'am, this this takes place when the temperature is lower, that I understood. But then if it is outside the body, because it needs a lower temperature, right? So like it will again come back
That
into
is
the
the
body
reason.
and it will again go out.
That is the reason. Since it is outside the body, and now the outer region, the outside environment of the body is all now, the temperature is now even more dropping down. So there is a tendency that the temperature in the testis will be much less. So at that time, the scrotal sac will contract and take the organ towards the body to get the body warm. So that the temperature does not drop much beyond, it only can maintain, it only drops something 2-3 degrees centigrade lesser than the body temperature, that's right.
Ma'am, but like most of the times the temperature that we have is like very less than the body temperature. It's like 30, 22 or something like 22 to 30. So like in that case, then it will be most of the time closer to the body, right?
And most of the time it is closer to the body, but if it is a temperature something around 18, 20, if it's a temperature something around 12, 14, then the contraction of the scrotal sac is even more. And when consumption of hot drinks or some exercise, the relaxation of the scrotal sac is even more. So let us complete that answer. What is it?
This happens when the outside.
Next point. Next point. The scrotal sac relaxes. The scrotal sac relaxes. The scrotal sac relaxes and takes the testis away from the body. Takes the testis away from the body. During vigorous exercise, etc. When body heat tends to rise.
So it is when outside temperature is cold and when body temperature is high.
Both the conditions you have to write. When body heat tends to rise, full stop. The testis is taken away from the body to maintain optimum temperature, for spermatogenesis. To maintain optimum temperature for spermatogenesis. Optimum temperature for spermatogenesis. Now, we are talking about this word spermatogenesis. What is spermatogenesis? You may be only asked to write down the word, name the following. Definition as such is not asked. What is spermatogenesis? So, when we are now talking about what is, name the method, name the process by which the diploid cells of the testis form functional male gametes. What is the process? Spermatogenesis. So write down that word, the next question, the last question, then we go ahead. Write down the next question. What is spermatogenesis? Remember this word. This word you'll have to remember to write correct spelling in that name the following section. Generally it comes on MCQ. The process by which diploid male cells of the testes are converted into male gametes, haploid male gametes. The process of formation of the male gametes is known as spermatogenesis. Process of formation of the male gametes is known as spermatogenesis. The process of formation of the male gametes is known as spermatogenesis.
Cells of testes are
Converted into haploid cells.
haploid sperms.
Sperms, gametes are always haploid. Gametes are always haploid. Now, talking about the testes a little bit more. Write two special features of the male gametes. Now, we have to move on to the concept of the testes again, little bit more to study. We were talking about the testes, we have studied a little bit about the gametes. Now, let us draw the structure of the male gamete and write down two special features. Now, what are the two special features? Write down your answer first. What are the two special features of the male gamete? Number one, it is a very small cell. As in comparison with the female gamete, it's a very, very small cell.
The smallest cell of the body and the female is the biggest cell of the body.
Very small. When we are, it's a very, very small cell. Right now, it is a very small cell with minimum amount of cytoplasm. Very small cell, microscopic, very small cell, minimum amount of cytoplasm. Number two, it is motile. Number two, it is motile. Now look into figure 13.4. I want you all to draw that same figure there. Draw all of you. I'll give you 5 minutes. Simple pencil drawing, neat, nice. This comes for 2 marks in your paper.
Which one?
Figure 13.4. Figure 13.4.
No other diagram comes from there.
From here, 13.4 and female reproductive system. Only these two diagrams. 13.4 and 13.4 and the female reproductive system.
13.2a.
13.2a. The rest of the diagrams, 13.2c, very common for your exams. Diagram based questions. 13.2a for drawing diagrams. We'll do this in the class. 13.2c, very, very common for your diagram study. 13.1, A, B, C. All the three very important. Now we are going to study C. First, let us finish with the gamete.
Or diagram based.
All these are diagram based. Diagram drawing only this two. Finish it up. 13.4 fast. Sharp pencil, neat diagram, labeling to remember to write all of them properly.
Well, this acrosome is made by micro.
नहीं, mitochondria is in the mid part if you see there.
It is near the Golgi bodies.
Acrosome is majorly cell organelle which is producing enzymes, which in Golgi body is producing an enzyme.
This produces an enzyme and that enzyme produces acrosome.
No, the tip itself is known as a acrosome, which is producing an enzyme which will help during the fertilization to dissolve the membrane. That enzyme is known as the hyaluronidase, which helps in the process of fertilization. Complete the diagram, all of you.
What is this?
Acrosome, nucleus, head. The white part, no, that's nothing.
We draw that.
No, no name.
Ma'am, the axial filament, does it show like that it has a presence of neuron or something?
It is just a tubular part.
What is the length of the tail matter?
No. Simple pencil drawing. Remember to draw the outline.
Ma'am, the mitochondria is like very
Spiral. Just draw some spiral thread-like structures. That is. This has to improve. Huh?
Ma'am, this is okay.
The diagram drawing has to improve a little bit. Make it prominent, nice, dark, outline proper. So straight away 2 marks.
We have to draw a detailed nucleus.
No, just like this. Just like this. Then label the parts.
And should be also like nucleus.
What
and
is
bracket.
this? What
Ma'am,
is this?
End.
What is this part?
acrosome.
Label the parts properly.
Arrow in the diagram.
What is this part? The nucleus. This part is the
Mite.
This part is the midface actually. What is given? Midface. Middle piece. This is
Mitochondria.
the mitochondria. Then this is the axial filament, which is here. And this part is the tail. This part is the tail. Outline should be neat. नहीं 13.4 I asked you to draw. Female later. नहीं, did we study the female so far?
सेल बनाना सिस्टम बनाना क्या
सिस्टम बना रहा है फीमेल सिस्टम
हाँ एग्जैक्टली
मैम
ये draw करना लग गया।
नहीं ये भी आप
यही करना है
ये
मतलब
draw
female
करना
system
लग
में
गया।
ये भी drawing है।
Yes.
अभी
अभी ये draw
But we'll draw that later.
Ma'am, but I already drew half of it. I don't have to.
Acha, you keep that, but the diagram of the sperm you draw fast. When you draw the diagrams, most of you, as you draw the diagrams, you draw it very casual, very simple, light, as if you don't have strength in your fingers. Draw neat, bold, nice. See there? See this diagram? See both their diagrams. This is how you should draw diagrams. See, but don't label with pen, Mark's diagram. See the diagrams that they draw. Straight away too. Okay? So neat, nice, bold, clear diagrams to draw. Most of you are drawing diagrams very, very casual. एक लाइन भी draw नहीं कर सकते हो अच्छी तरह से clear, neat, bold. So every diagram. Now that we are almost at the end of the syllabus, I'll be asking you to draw diagrams of the cell and all these things. अरे ये पहले करो ना. That will draw later. Draw this first. Okay. All done. So, this is good. Draw diagrams neat. You can take care. So while unnecessarily 1 mark will be gone there. Make it fast. We have to still study a little bit more about the structure of the testes.
Eraser.
In progress. काले का ओ हाँ ओके
कब ये हो गया
मैं वही सोच रही थी
क्या लिखा है?
आज इरेजर नहीं
Make it fast. Make it fast. Let us finish with this part.
NP6
Huh? Huh?
there is no NP6 just it's a middle piece NPs
नहीं.
D है ना NPs
How far? Syllabus in school?
Sensory organ.
sensory endocrine everything finished reproductive 20
For you all? All done? So this now next exam is full motion exam.
yes
After that, how many more prelims?
one and then course
And then in between they do revision. Chalo, now next.
I'm 1 minute every single name you have to know.
Major ones. I'll tell you which are the ones which are asked also. Not everything is asked.
This diagram is missing that is
Master's
That's not answered. हाँ, very good. Neat clear करना है, so straight away two marks are there.
और हाँ मैंने सोचा
How did
Just spiral thread-like structures to draw. Now, next, what are we going to study is this one. We are still to study a little bit more about the testes. So, about the first part, we have studied all these things are clear. Now, we study a little bit more about the testes, where looking into the internal structure of the testes, something more, the types of cells, etc. Looking into the internal structure of the testes, if we try to find out how would the section of the testes look like, the section of the testes would look like this. The section of the testes would look like this, where the entire testis is cut down into certain chambers. This is the longitudinal section of the testis. Just take a note of few terms.
like kidney.
The entire testis longitudinally when cut in a section has got chambers. What are these chambers called? Lobules. What are these chambers called? Lobules. So we are talking about the transverse section of the testis. We are talking about transverse section of testis. Which are some of the important terms, words, specific points to remember? The first I said lobules. How many lobules? Generally 200 to 250 lobules in one testis. Which are the lobules? These chambers. The entire testis, when cut in a section, is seen to have lobules, and in every single lobule, there is a tightly coiled, compact arrangement of tubules there. Tubes. So there are a compact in every single lobule, in every single chamber. There is a tightly coiled, compact arrangement of tubes. What are these tubes known as? Very, very important. What are these tubes known as? These tubes are known as the seminiferous tubules. These are known as the seminiferous tubules. What are seminiferous tubules? The extensive arrangement of fine tubular network inside the testis. Now, there are quite a number of them. The seminiferous tubules, there are so many of them. Now, the seminiferous tubules from each chamber, they all go and unite together, and then they result in the formation of broader ducts known as efferent ducts. Clear? So, this seminiferous tubules, they now go and meet, fuse, result in the formation of broader efferent ducts. But these efferent ducts, how many on an average? 16 to 20. So, from each of these chambers, what is happening? The fine tubular, compactly arranged seminiferous tubules, which are very thin, and these thin tubules, they all go and merge, fuse. They go and merge, fuse, and ultimately result in the formation of some broader tubes. Which are those broader tubes now known as efferent ducts, efferent tubes. Clear? This 12 to 16 efferent ducts, now they again coil and coil and coil, result in the formation of an extensively coiled tubular structure almost on the head of the testis. So what is this entire structure, extensively coiled tubular structure known as, which also partially forms a head-like structure, which also forms a partially a cap-like structure over the testis. What is it known as? This is now known as the epididymis. This is very, very important. Epididymis. So now again, chambers of the testis are known as
As
Nice.
like
Lobules. The tubular structures inside the lobules are known as seminiferous tubules. Seminiferous tubules fuse to form efferent ducts, and efferent ducts coil together to form epididymis, and epididymis, these 12-15 tubes again fuse together to form a broader single tube, and that is what is the vas deferens. What I said?
You said broader. No, in the diagram is thinner. That's what I was saying.
So this is a comparatively broad structure.
The vast difference is much thinner.
Much thinner. I said
Yes.
broad. Acha. So now this is generally a single tube now. So what are the connections? We are talking about seminiferous tubules, efferent ducts, epididymis, and then vas deferens. And this vas deferens is now moving upwards. Upwards where? Towards the abdomen. So now this is the entire testis. So this vas deferens will go and now merge with which part? The vas deferens will go and merge with which part? You'll have to remember the relations. The vas deferens will go and merge with the urethra. So we were talking about the section of this part, the testis. The section of the testis consists of lobules, and every single lobule having the seminiferous tubules. Seminiferous tubules are fine, thin tubular structures, and inside every single thin structure, the process of formation of the gametes going on.
Ma'am, like it is going upwards, right? So does it have any pumping or valves, or like the tail itself is enough?
The tail itself is enough. So when we are talking about, I'm saying that the process of gamete formation is here. All these words, seminiferous tubules here, efferent ducts here, epididymis, this is the epididymis, are you able to see? So if you get the labelings, this is the epididymis, and then the epididymis always fuse together to form the vas deferens. So this is the vas deferens. Now, what is the function of the vas deferens? We had already written earlier. Helps in the passage of the sperms from where to where? Testis to the urethra. Testis to the urethra. So now, a little bit more on the seminiferous tubules now. When we are talking about that, what about the seminiferous tubules till then? When we are talking about the section of the testis, this seminiferous tubule, every single tubule is carrying out the process of gamete formation. Every single tubule is carrying out the process of gamete formation. Now to understand that, let us try understanding that how would the section of the seminiferous tubule be like, because certain more types of cells we have to understand. So say suppose this seminiferous tubule we try to find out in a section. We want to find out the section of, it is a tube-like structure. We said this entire seminiferous tubule is a tube-like structure. Cut it in a section. Try to see how would this seminiferous tubule look like. So if this is the entire seminiferous tubule and we make a transverse section of the seminiferous tubule, we get to see that the entire inner lining of the seminiferous tubule has diploid cells. Now you are very clear. The moment I say diploid cells, you are clear. What does that word diploid mean? What does that word diploid mean? 46 chromosomes. So it is this entire, what am I drawing? I am drawing the section of a single seminiferous tubule. I am drawing a single section of a seminiferous tubule, trying to understand that these individual cells, where every cell has 46 chromosomes, what would happen when each of these cells undergo the process of meiosis and result in the formation of gametes? Yes, each one of them, they will form 4 functional gametes. Each cell of the inner lining of the seminiferous tubule, inner lining of the seminiferous tubule will have hundreds of such cells, diploid cells. Every cell is undergoing the process of gamete formation. Every cell is undergoing the process of gamete formation, and hence in the central cavity of the seminiferous tubules there are millions of these motile sperms, also known as spermatozoa.
And they're not motile. They become motile rapidly.
Partially formed, partially. So now, this, when we are talking about this inner lining of the seminiferous tubule, the cells are diploid. They undergo the process of meiosis. Now that they undergo the process of meiosis, they are in the process of formation of the male gametes. These male gametes, how many? 4 gametes per single cell of the seminiferous tubule. One cell forming 4 functional gametes, already I had shown you earlier. Now these gametes are the sperms, haploid cells, and in the process of maturation. Then something more to, what is that? Grabbers. Or your knees?
No.
No, not at all.
Huh?
No.
No, no. Tell me.
That definitely not.
Aditya? Okay. Wait, the other one today. Enjoying the
Yeah, he actually skips this.
Huh?
He skips the classes that are not ordinary.
Yeah.
That are not.
Ordinary time. It's like
Okay.
so he doesn't tell his mother.
अच्छा.
Bro, but message तो जाता है.
Are you sure?
हाँ भाई.
But उसको तो bio में करना है उसको.
Ma'am, but he wants to be
उसको क्या बायोमेडिक
Need.
है
है बहुत कुछ है भाई.
है बट उसके लिए नीड
He wants to become a neuro.
Definitely.
No, I'm not like I don't know if you want to become a neurosurgeon or a neurologist. So neuro something. Neurologist.
Now, when we are talking about this is one process. What are the 2 functions? What are the 2 functions of testes you have written? 2 functions. What are the 2 functions you have studied? One is formation of the male gamete, and now we talk about release of the hormone. Now, in between these cells, in between these male gamete forming cells, in between these male gamete forming cells, if you try finding out the entire section of the testes. So these type of small chambers are all over, and then they have some specialized group of cells here, some specialized group of cells here. And what are these cells known as? These cells are known as the interstitial cells. These are known as the interstitial cells or the Leydig cells. So now, when we are talking about the interstitial cells, what are their functions? Releasing the hormones. Which hormone? Testosterone. So 2 functions about the testes we already mentioned. What are the 2 functions of the testes? One is the formation of the gamete. The other is release of the hormone. So now, inside the seminiferous tubules, we come across arrangement of cells like this. So the 2 major types of cells, you are asked questions. Name the hormone releasing cells of the testes. Spermatozoa is the alternative term for sperms. Sperms or spermatozoa. Spermatozoa is the alternative word for sperms. So when you get this word, also try to identify. It's nothing, any other specialized cell that we are talking about. Spermatozoa is the same for sperms, right? It's just the alternative word for sperms. It is just the alternative word for sperms. So now, talking about the 2 types of cells. Name the hormone secreting cells, always to remember, the Leydig or the interstitial cells. Now, next is the function of the epididymis. Let us try understanding here. The sperms are moving. What is the direction of movement of the sperms? From the seminiferous tubule. Next way. From the seminiferous tubule to the efferent ducts. When the gametes are in the efferent ducts, they are still not motile. They are partially mature. The cells are still partially mature. Now that the gametes are in the epididymis, and since the epididymis is extensively coiled, this is the head of the epididymis, this is the body of the epididymis. The epididymis is an extensively coiled tube, so these gametes are present here for a very long time. It is during this period when this single cell gains its motility. When the single cell, so name the part of the male system where the male gametes become motile. They become motile in the epididymis. Are the male gametes motile in the seminiferous tubule? No, they are formed, partially mature. They gain their motility in the epididymis. And now that they are in the vas deferens, they are constantly moving towards the urethra. This male gamete, now that we talk about that, now what is the function of the vas deferens? Or the vas deferens is carrying the male gamete way to the urethra. So talking about the sperms now, what are the features of the sperms we had come across? Motile, very small, microscopic. For how long does it remain functional? It remains functional only for a period of 2 to 3 days, meaning we would say that the male gamete is having a lifespan of 2 to 3 days. The male gamete has a lifespan. What is the meaning of the word lifespan? The time till which the cell is functionally active. So this male cell, the male gamete, can go for the process of fertilization in this period of time, 2 to 3 days. If not, then the cell will start disintegrating and dying out.
In the reproductive system for 2 to 3 days only.
It is present, it is alive in the male reproductive system, or if it has entered into the female reproductive system right from its release, from its formation, it will remain active for a period of 2 to 3 days only. So during this period, if fertilization, then the zygote.
If it is still present in the epididymis, and then how long will it live?
It will not remain in the epididymis for a long time. It has a specific course of movement. It will be here for a few hours, and then after that it will keep on moving. But because there are millions of cells there,
At what interval like it is created? Like 2 to 3 days, every 2 to 3 days you are at it.
what is created?
The cells.
The male gametes. The male gamete can be created at an interval of few hours also.
But then it is created in an interval.
The male gametes can be formed at an interval of few hours also.
Not every second.
No. Not every second. At an interval of few hours, because now say suppose at one particular period of time, the male body has produced adequate amount of gametes. For a short period of time, the body will not be able to make gametes because these gametes will be running out of the body. The gametes will be all cleared, lost from the body for
5 hours. Sounds like
on an average 4 to 5 hours. Once the gametes are formed, immediately within half hour again the body cannot prepare adequate amount of gametes. Because in case of the male body, both the testes undergo the process of gamete formation together. All the cells undergo the process of gamete formation simultaneously. So if I talk about the testes in the male body, both the testes at a time produces millions of gametes. So after it has produced an adequate number of the gametes,
like 20 million to 40 million.
2 lakh
One ejaculation.
2 lakh 4 lakh.
Like 20 million to 40 million.
200 to 400 million.
20 million.
2 million. So now that we are talking about that this type of,
20 million.
when we are talking about this,
200.
this particular process of gamete formation always at a particular gap. Yeah. Now this is still faster in the female body. It is at a gap of almost 30 days. In the female body, the process of gamete formation is at a gap of 30 days. In the male body, the gamete formation is at a gap of few hours only. Yeah. Now, next is, we talk about this. Next, we try understanding that now that the gametes are carried, the next part is what? Now that the gamete is carried by the vas deferens, and now that the gametes are released where? Into the urethra. Now the gametes are released into the urethra, surrounding the urethral tube and the junction where the vas deferens meet the bladder and the urethra. At this particular region, there are some more glands. At this particular region, there are some more glands, and these regions or these glands in the male system, they are known as accessory glands of the male system. So what were the primary organs we had studied? Primary organs, those four parts. The testes, the vas deferens, the urethra, the pelvis. These are the primary organs of the male system. Then we studied about the section and so many essential parts and the types of the cells. Now the last and the third segment of this topic is knowing about the accessory organs. So which are the accessory organs? Now talking about the accessory parts, let us try understanding what are some of the accessory glands of the male system. Now what does the word accessory mean actually?
They support, but they are not.
Supporting, correct.
Not directly.
Supporting, not very much necessary, but can enhance the function, can enhance the ability. We wear accessories. Why do we wear accessories? So that our daily functions can be even more smooth. We look smart. We sometimes. So that is to enhance any type of a function. Same like that. When we are now talking about the accessory organs, what are the accessory? Yes. What is it?
That's good part.
What is it? What is he saying?
Aditya was buying his these.
So that is what I was suspecting.
He's faking, Arju's faking.
Okay.
Huh?
I will give a big blow on your face.
Ma'am, the seminal vesicles, how are they accessory?
They are accessory.
So without them, we can.
The efficiency, the chances of fertilization will become very lesser. Maybe lesser. Will become lesser. We'll discuss this now. So we are saying that the male system has got some accessory glands, additional glands, some extra glands which can enhance the release of the. Now, these accessory glands has no role in the formation of the gametes. Formation that we have studied already. The cells and the hormones and etc. We had studied already. Formation में these accessory glands have no role. Always to remember, these accessory glands, they have no role in method of formation of sperms. Method of formation of sperms is known as spermatogenesis. So they have no role in the process of sperm formation, but they play an important role in the release of the sperms from the male body. What are those glands now? The first is the seminal vesicles. What about the second? The second is the prostate glands. What about the third? The third is the tubers or the bulbourethral glands. Now let us talk about this. The three types of glands. Now the function is very, very important and the location. That is what you are asked in your exam. Let us talk about the first one. See the location. When we talk about the location of these glands, the first one, seminal vesicles, are you able to see? Seminal vesicles. Mark in your textbook also the location. It will just take 5 minutes more. Mark in your textbook the location also for each of the three glands. You are definitely specifically asked questions on this. Seminal vesicles. What is the location? It is at the junction of the vas deferens and the urethra. It is at the junction of the vas deferens and the urethra, the seminal vesicle. What is it doing? What is it releasing? An alkaline secretion. What is it releasing? This seminal vesicles. What is it releasing? The seminal vesicles are releasing an alkaline secretion. It is releasing an alkaline secretion, and this alkaline secretion, what will be the job of this alkaline secretion? Provide nourishment for the sperms. It provides nourishment for the sperms. That's a major function of the seminal vesicles. So this seminal vesicles, these are secreting some alkaline secretion, pouring it where? Into the urethral tube. What is the function? When you are asked in your exam, write down the function. Students would write release alkaline secretion. You will not get anything for that because all the glands are producing alkaline secretion. And function specific here for all the three. Okay. What is the function of the seminal vesicle secretion? Release alkaline secretion, provide nourishment. Provide nourishment to the sperms. Why nourishment necessary?
The same as prostate gland.
Prostate gland
Into
generally.
the urethra.
Prostate gland generally reduces the acidity of the female reproductive tract. That is different. The function of the prostate gland is different. So the secretion of this gland provides nourishment to the sperms, and the sperms should remain motile for a long time. They require nourishment. Talk about the second. What about the prostate glands on either side of the urethra? Mark the location. Either side of urethra, that is the location of the prostate glands. This prostate gland function, when you are asked, what should you write? This also produces an alkaline secretion. That is the same thing. This also produces an alkaline secretion, but this reduces or neutralizes the acidity of female reproductive tract. I write it short. The female reproductive tract. Remember the functions are very specific. You get differences also. Now, this prostate gland reproductive tract. Acidity. Alkaline secretion, the prostate gland is also releasing alkaline secretion. But this alkaline secretion is not for providing nourishment basically, but to release or neutralize, reduce the acidity of the female reproductive tract. Now, how the acidity of the female reproductive tract? This secretion is in the urethral tube. The urethral tube has the sperms also. So the sperm and the secretions together of the accessory glands, that mixture is actually known as the semen. So what is the definition of the word semen? It is the sperm and the mixture of all these three glands.
Three glands, all three, or only the two?
All the three. So the mixture of the sperms and the alkaline secretion of all the three glands, which is in the form of a milky secretion, that is what is the semen. So you get questions that are mainly suffering from problems in the prostate, or a male has removed the prostate gland. Will this male be capable of participating in the process of fertilization?
Yes.
No.
And then how is this an accessory gland?
Because now that a male, if by chance, due to certain disease or age, when the male has removed the prostate, the secretion of the prostate is no more releasing. The semen does not have the secretion of the prostate glands. So when this semen enters the female body, the female reproductive tract is extremely acidic, which kills sperms in large numbers. So this secretion actually increases the chances of fertilization.
But that can be
If this secretion is very less or not there, chances of fertilization becomes much lesser. So this is the concept that you'll have to write down.
It can be treated, right?
The treatment is different. So that is the reason when we are talking about after a particular age, I talk about a male at the age of 50, male at the age of 55. Will he generally have a high chance of participating in the process of fertilization? No. Give a reason. The prostate is not releasing adequate amount of alkaline secretion, so the acidity of the female reproductive tract cannot be neutralized. Okay? And the last is very simple, the Cooper's glands, the bulbourethral or the Cooper's glands. Mark in your book also. The Cooper's or the bulbourethral glands, location on either side of the urethra. Function, This also releases alkaline secretion, but the secretion is majorly helping in lubrication, meaning does not allow the sperm to stick to the walls of the urethra. If the urethra tube is dry, the cells will stick, they will die out. The objective is release out from the male body, and if the process of fertilization has to be thought, it has to enter the male body. Inside the male body also to remain motile for at least a specific period of time.
Ma'am, what is the fluid it releases?
Semen,
No,
the mixture.
the bulbourethral.
All of them are producing alkaline secretions. All of the three. So that's the reason. That's the reason whenever you are asked to write down the function, not to write alkaline secretion release. Functions, lubrication or neutralizing acidity or providing nourishment. These are the functions to write. Now the next class will be studying about the female system and the process. What is it?
कितना देर लगा
If you draw this diagram, practice karo, you get full marks.
वापस दर्ज है
करीब